Hello, everybody. I'm at the Institute for Research in Biomedicine, and I will talk briefly about a highly interdisciplinary project that has combined physics, biochemistry, and medicine in the field of protein dynamics. Let me start with a metaphor to illustrate why dynamics is relevant for protein function. As you can appreciate, not only the shape, but also the coordinated motions of a horse seem designed by evolution to run. So until fast photography techniques were developed, all representations of running horses were completely wrong. And in the case of proteins, proteins like horses also have shapes that have evolved to perform certain motions that are required for their function, but we still don't have fast cameras, and usually we only have a couple of static photos of a protein that come from experiments. So the only way we have to reconstruct the motion of a protein is to use the laws of physics and do some kind of simulations to get a trajectory. So protein simulations can be done in two ways. Atomistic way is like a very detailed painting, such as this portrait in which you can almost see the eyelashes of the girl. And the coarse grain view is like a Velázquez painting that only gives you like an overall impression with very coarse strokes. In an atomistic simulation, you consider all the atoms and all the interactions between them. And a coarse grain simulation, you only take into account the backbone of the protein structure, pack all the forces into a simple function, such as an elastic spring. And this allows you to increase the speed of the calculations, but at the cost of some accuracy. However, combining both approaches, it's possible to get powerful insights into the mechanics of proteins, such in this case, the HER1 receptor. HER1 is a protein that plays a gain function in cells, and that is mutated in a number of tumors, such as brain cancer, glioblastoma. This protein acts as a molecular switch between two structural states that are known experimentally. The closed state, in which the molecule is inactive, and when a growth signal appears, it triggers a change in shape towards an open configuration that is active and starts cell division. It seems that this process fails in cancer, and some brain cancer mutations are able to activate the protein without the presence of any growth signal. So mutations in cancer seem to be clustered in particular sites of the structure. So we first applied a coarse grain approach, an elastic network model in which the protein structure is represented as a network of amino acids connected by springs. And we mimic the effect of mutations by perturbing this network. And we observe that the protein has a closing and opening equilibrium. And in addition, that changes that are hitting at regions that act as hinges between different modular parts of the structure can have really the potential to change the dynamic of this protein. So we hypothesize that probably mutations are clustered in these sites and could be like adding some extra oil in the hinges to facilitate the opening and closing of the structure. So we took one step further, and we confirmed this idea with microsecond-long atomistic simulations. And we observed two very different movies. The normal protein with the growth signal opens as expected, but the mutant protein seems to be able to open alone without the need of any signal. And in addition, it does so in a completely different way that leads to a structural state that has never been observed before experimentally, but that seems to be the hallmark of a number of tumors and to explain a great deal of the HER1 biology. So the main idea of this project is that really the proteins really move, and these motions are the key to understand how they function and how they misfunction in some diseases, and that by using physics, it's possible to give a rational explanation for the location of mutations in cancer target proteins, and also it's even possible to reveal states of proteins that are inaccessible to experiment, but that are really important to understand how they function and can help develop better treatments. So thank you very much.